Okay, I think we're ready to start. Okay, so all our speakers in this session are going to be telling us various aspects on woody vegetation dynamics. And our first speaker is going to be Dr. Naledi Zama. She is associated with the University of KwaZulu-Natal as well as Sol Plaatje University.
Afternoon, everyone. My name is Naledi Zama, and I will be presenting to you today some work that we have conducted at the Munywana Conservancy. Okay, so savannas, as we know, are ecologically and economically important. They support biodiversity, livestock, and they cover about 20% of the Earth's surface. However, these savannas can be encroached by a lot of woody species. This means that the woody density may increase, the density, the biomass, and that all affects the structure and the functioning of these savannas. Now, woody encroachments can either be very positive or negative. Positive in the sense that some nutrients can be added onto the soil, whereas in other instances, the nutrients can get depleted and grass fuel load or biomass can also decrease. Now, some reports indicate that encroachers can form islands of fertility. So what is this? So you find a case where beneath a bunch of trees, the nutrients, moisture, and soil organic matter accumulates so much that compared to the outer space, so you find that those areas are very rich compared to the outer spaces. So for my study area, which was the Munywana Conservancy, this conservancy is reported to be very heavily encroached, specifically by two species. So these woody species are Vechelia nilotica, commonly known as scented thorn, as well as Dacrostachys cinerea, commonly known as sicklebush, and also known as ugagane in isiZulu. Now, both of these woody species are nitrogen fixers. So this means that they are able to form symbiotic relationships with soil microbes, and then they are able to convert nitrogen to a form that can be utilized by plants. However, this process also requires the use of phosphorus. So you find a case where phosphorus levels in the soil can also decline as a result of this. Which brings me to my hypotheses. So for the sake of this presentation, I'll only just be sharing 4 hypotheses. So the first one looks at how grass fuel load is expected to decrease after a threshold of 2,500 trees. The second one is woody density could change the soil organic matter, but we're not sure about the direction because a lot of reports indicate increases in soil organic matter as well as decreases or declines, and others report no change at all. The third hypothesis is that increasing woody density will lower the soil pH, mainly because of the acidifying effect of the nitrogen fixing species. And then the last hypothesis is that these encroaching nitrogen fixing legumes can increase the topsoil phosphorus because they might have the ability to access the deeper soil P layers, or the P levels in the soil will decline because these nitrogen fixing species demand phosphorus to undergo biological nitrogen fixation. And this will be especially true in soils that are very clayey. Okay, so this study was conducted, as I said, on the Munywana Conservancy. There were 27 monitoring sites. So if you look at figure 1, that little dog there, that's the study area. And all of the sites were found within one of the 5 vegetation types, which were Maputo land, pallid, sandy bushveld, the southern Lebombo bushveld, Dembe, sandy bushveld, western Maputo land, clay bushveld, as well as the Zululand low veld. Woody density was collected using the point center quarter method. And for this study, we only looked at trees that were at a height less than 2 meters because these are the trees that are most encroaching in the area. So it only made sense for us to focus on these particular species. And then for the analyses, I focused on total woody density density of Vegetation nilotica, and the density of Dicrostachys cinerea. All of the soils were collected around the similar time. It was only topsoil which we focused on. And for grass field load, they used this meter to determine grass field load. And then in terms of the analyses, we use a combination of linear mixed models, generalized linear mixed models with segmented terms, and all of that to analyze the various hypotheses. So, okay, what are the results? The first hypothesis. So here I had an initial guess of 2,500 trees per hectare. That was my initial guess, that will be my threshold. That is where we will see a decline in the grass field load. Of course, with data, especially data that's collected on monitoring sites, not the case. The model itself estimated a breakpoint of 6,384 trees per hectare. However, our segmented regression found that neither the slope before nor after the threshold showed a significant difference. So in other words, there was no effect of total woody density on grass field load. Hypothesis 2, again, here we see no significant relationship between the woody density and the soil organic matter, right? So it looks a little bit like the slope is increasing. Yes, the slopes are increasing, but if I spread out my graphs more, you can see that that increase is very slight. Also, if you look at the beta coefficients, let's see, these ones, so these beta coefficients here, you can see that that increase is minute, it's small, it's increasing the slope, but the slope is not significant. So again, just because your model was not significant doesn't mean it was useless and you need to go cry in a corner somewhere. No. What it means is maybe look at another aspect. So here I decided to look at my random effects to see if, did my random effects have an influence or my results in any way? So what I found is that vegetation type, which is my random factor in all of my models, seemed to be important. So if you look at table 1, I show you there the ICC value. ICC is the intraclass correlation coefficient. This is a value from 0 to 1. The higher the value is, it means that your random factor, or in my case, the vegetation type, is important in explaining the total variance in the response variable, which is our soil organic matter. So it seems that for the Dicrostachys cinerea and the Vegetation nilotica models, very low ICC values. So we're just going to focus on the total woody density. The value is 0.60, which is 60%, which is quite high. And if you look at figure 5, I just drew that plot so you can just be able to see what I mean. Sites in Tembe have very low soil organic matter. Sites in Zululand Lowveld, much higher. And you can see that how they differ. So that's the point I'm trying to put, is that, okay, our model was not significant, but our random effect does indicate that vegetation type influences soil organic matter in terms of total woody density. Hypothesis 3. So here, this is for the soil pH. Again, we see slight increases in the beta coefficients, but woohoo, it's not significant. So as much as it seems as if it's increasing, that increase is not significant. So there's no significant relationship between the woody density and soil pH. Hypothesis 3, again, ICC values. Vegetation type seems to matter more for pH in terms of the Vegetation nilotica density and total density, and not so much for the Dicrostachys cinerea density. Next, the last one. So this is the more interesting result. So this is where I'm trying to determine whether soil phosphorus levels increased or decreased and whatnot. These are the results of the main effect of total woody density on phosphorus. We see a significant decline, yay. So here we see that the decline is significant. And for figure 9, we also see a significant relationship between the soil clay as well as soil phosphorus. So we see that the phosphorus levels are increasing. But again, these are just the fixed effects, the main effects. What is more interesting is the interaction. So we did find a significant interaction effect. This was for the relationship between Vegetation nilotica and soil clay percentage in predicting soil phosphorus levels. Because an interaction is very weird to plot, you have to find ways to plot it in a way that other people can understand. So what I decided to do here is split up my clay into two parts, high and low. And this is relative to my data. So the 14.7% is the median of the clay percentage. So clay that had a clay percentage of 14.7 or more was considered high, and then sites that had a clay percentage less than 14.7% was considered low. Now, how we look at this is that we look at the direction here. So in soils that have a high clay percentage, having a high vegetable density in those areas means that you'll most likely have less soil phosphorus. In soils that have a low clay, it's likely that increasing density will increase the soil phosphorus levels. Okay. And then in terms of the discussion, so the main thing is that in this study, there were two vulnerable vegetation types that are considered vulnerable in terms of conservation efforts: Zululand no-fault and Western Maputo land clay bushveld. These, because of this reason, they always need to be monitored because they are currently very vulnerable. So that's the first thing. Secondly, there are concerns for sites that are in Tembe, sandy bushveld, because all of these sites had a pretty low pH, low phosphorus levels, low organic matter, but the density, the woody density was high. So these should be priority sites. Thirdly, the Chelia analytic density and the clay interaction was important. In soils with higher clay and higher density, you get less phosphorus, which indicates a self-limiting feedback. And then in sites that have clay percentage less than 14.7%, and you have higher density, you get higher phosphorus levels, indicating sort of reinforcing dynamic in those areas. And then for the fourth point, Dacrystachys cinerea showed no significant interaction with clay. Some reports indicate that this species is able to access deeper soil phosphorus levels. Maybe that's the reason. Maybe it's an issue with our data. Who knows? And then for point 5 is these results are results from a single point. So it's like a snapshot of a particular time. What needs to happen is they need to go and re-survey the same plots again, or the same areas, so we can see if there's a change, like a temporal change, because just looking at one point in time might not tell you much. Yes, we are able to maybe see what's happening at this point, but I feel like reanalyzing, resurveying and reanalyzing to compare the two times in 2 years will provide useful information. I'm running out of time, so I'm just gonna go quickly. So I'm concluding now. So because the study was more about helping the Conservancy make management decisions and whatnot, I thought for the conclusions, it would only make sense to put something like this. So these are known as quadrant plots. Briefly, if you just focus on the plot that is shaded, that's known as the quadrant 4, which is the quadrant that includes all the sites that are vulnerable. These are the ones that have high woody density, low phosphorus. If they want to know right now which sites should we focus on and go check what's happening there, or maybe start burning more frequently or whatnot, they should look at this and decide, okay, we should probably look at sites in the Tembe, so these green ones. These sites, all of these sites are the ones that are considered vulnerable in relation to the results of this study. Okay. And with that, I'd like to acknowledge the Phinda Private Game Reserve and the University of KwaZulu-Natal and everyone who was responsible in collecting the data, because I did not collect it, I analyzed it. All right, thank you so much.
Thank you, Naledi, for the great talk. Do we have any questions for Naledi? Yes, Susie?
Thank you. In reviewing the literature about the effects of trees and woody encroachment on things like soil P and so on, this island of fertility effect, did you look at the effect of tree size? Because I've sort of been through quite a bit of that literature over the time, and it seems that large trees, which are also old trees and big, tend to have those positive island of fertility effects. but dense stands of small trees tend to have negative effects because they haven't got the age to be pulling up the nutrients and enriching the soil. And then often their negative effects in cutting out rain and so on and shade tend to be more negative. So that's just something worth considering. I don't know if you've looked at it.
No, I didn't. I think the main reason why I didn't look at that is because we initially said, because most of the species that are encroaching are in that height, let's just stick to that. But that's a valid point. I think I should definitely read up on this, my tree heights, and see what literature says in terms of the negative effects or, yeah. True. Thank you, Susie.
Sorry, we've got another question from Mulindo at the back.
Of course.
Sorry, Naledi. Yes, I'm interested in checking whether did you then compare with an adjacent unencroached site to see if these effects are there?
So as I said, the data was not collected by me, it was already collected data. The team that works in the Conservancy only went and sampled sites that they feared were a problem. So they would not need to go collect anything in a site that was not encroached. So no, we did not compare with an unencroached area.
Okay, thank you. I think that's about enough time. I am now going to hand over to the rightful chair of this session.
Okay, hi guys, I am the chair again. So, our next speaker is Dr. Bukho. She will be telling us about woody vegetation dynamics in abandoned croplands in the semi-arid Savanna rangelands in the Limpopo Province. Yeah.
Thank you, Naledi, and good afternoon, everyone. As I've been introduced, my name is Bukho Gusha. I'll be talking to you about the work that we're currently doing in the Limpopo Province. The work is titled Investigating Woody Vegetation Dynamics of the Abandoned Croplands in the Semi-Arid Savanna of the Limpopo Province. So cropland abandonment is defined as the cesarean of active cultivation on land that was previously cultivated. And this has only not been reported in the Savanna, semi-arid Savanna regions, but it has been reported widely. And there's quite a lot of drivers that are reported to be the major cause of cropland abandonment in these areas. Those includes your climate change, the social and the economic factors, and some of the land use practices. So this trend has been especially pronounced in the rural and communal areas because of the socio-ecological and economic factors, like I said, and there's been some of the provinces, which is the rural provinces, where this cropland abandonment has been reported to increase between 0.8 to 0.28% annually. And these include the Limpopo Province, the Eastern Cape, and the KwaZulu-Natal. And these are one of our provinces in South Africa. So once these lands become abandoned, they lie fallow, and some of the natural ecological processes start to reshape the landscape, where we start to see some of the early pioneer species starting to emerge, and then it continues until some of those climax species starting to emerge. So one of the most significant processes that emerge as part of the processes includes the woody encroachment, which has been, most presenters have spoken a lot about it. And this shift has a wide-ranging implications for not only the woody diversity, but the herbaceous diversity, soil fertility, water cycle, fire regimes, and the local livelihoods. So there's been quite a number of studies that have been conducted to look at woody encroachment, and there's been a lot of speakers that I said that have been talking about the studies that have been conducted on woody encroachment. But these studies have mostly looked at woody encroachment in relation to fire suppression, herbivory, and land use changes. And the focus has only been on the intact rangelands, or they will focus on the protected areas. And there's been relatively limited information which has been reported on the relationship between the abundant croplands and bush or woody vegetation development, especially in the communal lands. So understanding how and why woody vegetation returned to this range landscape is very crucial for informing sustainable rangeland restoration, especially for communal areas. So we asked one of the questions that we wanted to ask and understand is how do these vegetation dynamics compare to a communal rangeland that have not undergone intercropping? So we looked at, just to bring in the context of where we wanted to look at how these woody vegetation dynamic compare to the natural rangelands. We looked at the the satellite images for the area where we are going to be collecting our study. And then we checked how is the extent of land abandonment in relation, cropland abandonment in relation to woody encroachment or establishment. Then we conducted the study in one of the villages in the Limpopo, Munywana local municipality in Limpopo, where we looked at the natural rangeland, and we compared that with the abundant cropland which was adjacent to it. So the study area is in the semi-arid Savanna, receiving between 400 to 600 millimeters of rainfall, and falls in the Munywana Plateau bushveld. So because we're only focusing on woody vegetation, so we established 6 10-meter by 10-meter plots where we collected all the woody plants that we found inside, and we identified at a species level. We looked at the tree characteristics such as the height, the lowest risable height, the canopy diameter, the short canopy. Then we estimated the crown tree cover using the following formula, and we look at the Shannon-Wiener diversity index. We look at the mimic richness index, and then we also look at the evenness. So in terms of our results, we find out that the natural rangeland had more tree density than the abundant cropland. But what was interesting was that in as much as the natural rangelands had higher tree density, we found out that the trees were taller. Were shorter than those that were found in the abundant cropland. And for the first one, we thought maybe these undisturbed areas may support more denser woody cover because there's not been anything that has been done on it. However, the taller trees may potentially reflect the competition for light and space due to lower tree density. And maybe even though we are still waiting for the soil analysis results, we thought the tree height, which is higher in the abundant cropland, may be related to the previous cultivation or the previous nutrient deposition that has happened due to cultivation. Then in terms of the tree crown area or the tree cover, so the abundant cropland, like I said, it had shorter trees. It had lower density but high trees, and those trees had bigger canopies compared to the natural rangeland. Then in terms of the composition, we find out that the natural rangeland had more of the distochite scenario compared to the abundant cropland. And then we found out that Vachelia robusta was mostly found in the abundant cropland. And Vachelia robusta is one of the native encroaching species, and it has been reported to encroach abundant croplands. And lastly, Vachelia carew, which is it's hidden by the picture, by the video. But the Vachelia carew was also reported in the natural rangelands. Then in terms of the species richness and diversity and evenness, we found out that there were no significant difference between the two sites. However, in as much as there's no significant difference, we can see that the mean species diversity and the mean richness was higher on the natural rangeland compared to the abundant cropland. And we think maybe to try and push these indexes up for the abundant cropland would be better if those lands can be properly managed, because there's kind of no management that's taking place. Then in conclusion, these findings demonstrate the importance of land use history in shaping the savanna vegetation, as the savanna woody vegetation dynamics, because there's no management practice that has been employed to manage this. Then we assume that, and because I also spoke about maybe the nutrients that were from cultivation, maybe the one that would drive the development of woody encroachment. So while the abundant croplands are undergoing natural regeneration, the current species composition and structure suggest that these areas are still in their early stages of succession, and they are, as they are dominated by a few robust colonizers. And over time, and with appropriate management, this system may move towards the greater diversity and structural complexity. However, if then there's no management practice that is being implemented or is being introduced, they may also become locked in a low density rate, as we have seen in the previous slide, and they will continue to be dominated by encroacher species. And these encroacher species are likely to reduce ecosystem services and alter the fire regimes and forage availability. And I thank you.
Thank you so much, Dr. Bukho. Due to time, we do not have time for questions, but I think if you want to ask questions, you can ask Dr. Bukho at a later stage. So thank you.
Thank you.
I'd like to introduce the next speaker, who is the last speaker for this session, Professor Peter Scogings. He will be telling us about assessing browse quality. What should we measure?
Good afternoon. I'm basically here to share some of my thoughts with you about what we should be measuring when we're assessing browse quality. These are thoughts that have developed over quite a long time. We know that large mammal herbivores in African savannas, in particular the mixed feeders and the browsers, have significant impacts on woody plants. And those impacts reveal themselves pretty quickly over time at landscape levels where you can very shortly after increases or decreases in herbivore populations, you can see structural changes in the vegetation. But what is not so clear is any species changes that take place in these systems. And the kind of scenario that I'm thinking about where what I'm going to say could be applicable is in situations where a defaunation event happened 100 or 1,000 or 10,000 or 50,000 years ago. And now there's movements to apply rewilding to processes to get large herbivores back into these systems where they've basically been extinct for a very long time. Obviously, during that time since extinction, the composition of the vegetation has changed from what was 100 or 10,000 years ago or whatever. But it would be useful to be able to predict which species of woody plant are going to persist and which are less likely to persist as a result of introducing large mammal herbivores into these systems that have been defaunated a long time ago. So the kind of traits that we're interested in is the traits that reduce the degree of herbivory by deterring or repelling herbivores or by reducing herbivore abundance, for example, by reducing herbivore fitness or survival. And these are the kind of traits that would enable certain woody species to persist under increased herbivory conditions. We've known for a very long time, some 40 years or so, there's substantial evidence to show that spines on thorn trees are a deterrent to medium-sized browsers and mixed feeders. They're not very, they don't really do anything for the mega herbivores. The elephants and rhino basically ignore them. But for the smaller herbivores, impala, kudu, etc., these are important deterrents to their foraging behavior. They basically reduce the foraging rate, they reduce the short-term intake rate of food, so it takes longer for the animal to consume what it needs to consume. More recent studies have showed furthermore that the structural complexity of the plant does the same thing. The more complex, the more structurally complex the plant, the harder it is for the animal to gain what it needs. And these structural traits that defend the plants, like so many other plant defenses, can be enhanced as a result of increased herbivory. So spiny plants become more spiny, structurally complex plants can become more complex, further reducing the ability of the herbivore to get what it needs. One of the things that remains to be determined is the extent to which these structural defenses reduce herbivore fitness, reduce animal performance, and ultimately affect population dynamics. That's completely unknown as far as I can see. Dave Augustine a few years ago did a very thorough review of relevant literature, mainly experimental stuff in African savannas, to try and identify traits that enable certain woody species to persist through the browser filter. And his conclusion, summarized in this conceptual diagram here, was that the plant species that produce certain chemical compounds are the ones that are most likely to persist through the browser filter. However, unlike the structural defenses, the condensed tannins and total phenols, which are the chemical defenses that everyone looks at and are the most well-studied chemical traits, there's the evidence for those being enhanced by increased herbivory is rather scarce. It's not as clear as it is for the structural defenses. However, and this is another thing that we've known for some 40 years or so, is that condensed tannins reduce digestibility and they reduce protein availability. This has been shown in numerous studies. Also, several studies in different parts of the world have shown that certain chemical compounds can indeed reduce herbivore fitness. They can reduce reproductive success in the herbivore population. And in certain cases, the chemical compounds in the woody plants can in fact regulate population dynamics of the herbivores. In a series of studies that we did over a course of about 15 years at 3 different sites, varying in vegetation, soil, and rainfall, in which we were trying to establish whether it was the structural characteristics or the chemical characteristics that were determining diet selection of domestic goats both free-ranging and confined, all in experimental conditions. And we found consistently that the tannins were not deterring the goats from feeding on them. In fact, the goats could get a much higher intake rate from the non-spiny species. So the spiny species were deterring them, the condensed tannins were not deterring them, and the goats preferred to eat on those kind of plant species. However, in the same set of studies, we also found, as we would expect, is that the condensed tannins have a negative effect on the animal performance, weight gain, nitrogen availability, etc. And that's the kind of thing that we would expect. So the thing here is that the structural defenses are interfering with short-term intake rate, bites per minute, or intake per minute, or whatever. And the chemical defenses are interfering with the long-term intake. So kilograms of dry matter per day, for example, or per week or something. Now, one of the things that I want to emphasize here is the problem with using general assays for determining condensed tannins and total phenols. For example, the acid butanol assay, which I'm sure many of you are familiar with, and some of you in the room have done these assays yourselves. And these are problems that we've known for a long time, but in my view, no one is really paying attention or sufficient attention to these problems and how to get around them and what we should be doing. So one of the main problems with the acid butanol assay is your results, your quantification of condensed tannins depends entirely on the tannin that you select to use as your standard for quantification purposes, for converting the absorbance values into percentage dry matter of tannin. So if you're using an appropriate standard, you can get a good approximation of the quantity, but you still don't know anything about the quality of those tannins. You don't know exactly what those tannins are. This study here illustrates the consequences of selecting the wrong standard. The black bars there are quebrachotannin. It's readily available commercially, but as you can see, it way overestimates the actual tannin content in the gray bars there. The white bars are sorghum tannin. It's get a much better approximation of the real tannin content. However, as far as I know, it's not readily available commercially. You basically have to know someone who's got some stashed away somewhere who can give you some to use. Alternatively, you can look at this particular paper here, and there's an equation in the paper that converts absorbance into sorghum tannin equivalents. Those gray bars are derived by extracting and purifying the tannin from each of the study species, and that is the best thing to do, and use species-specific tannins, and we should be doing more of this. Condensed tannins are an end product of the flavonoid biosynthesis pathway together with the anthocyanins, which are the ones that cause pigmentation in plant tissues. They both have leucocyanins as precursors, which is why I'm rather skeptical of Makar's equation for converting absorbance values into leucocyanin equivalents, because the leucocyanins could either become anthocyanins or they could become condensed tannins. The other two groups of flavonoids compounds that I've highlighted there, the flavones and the flavonols. The flavonols, I'm going to say a bit more about them just now, but the flavonols are derived from an intermediate step in the biosynthetic pathway. The flavones are derived much earlier in the pathway. The point that I want to emphasize here is that the condensed tannins, because they are polymers, they are high molecular weight phenolic compounds. The other groups of flavonoids compounds are low molecular weight phenolic compounds, and that's an important distinction. In this study here, we profiled with HPLC techniques, we profiled the flavonoid composition of 6 species representing 2 broad functional groups of woody plants in African savannas. And you can see very clearly in those chromatograms that no 2 species are alike. When we look at this in relation to what we know about diet preference of large herbivores in general. You can, there's an interesting pattern here, and I think it's a significant discovery, is that the less preferred species are characterized by flavonols, the more preferred species are characterized by flavones. In a follow-up study, we found the same result, looking at a suite of about 70 woody trees and 70 tree and shrub species across Southern Africa. In relation to diet selection by black rhino, we found the same pattern where flavones are a characteristic of the preferred species. The question here is, how does a browsing or a foraging browser or mixed feeder know that the plant that they are testing or about to eat is full of flavones or flavonols? We're not really sure how they can detect these groups of compounds. However, an answer to this question may lie in looking at the volatile compounds. And there are several studies that have been coming out recently showing that the volatile compounds are really important in determining diet selection. Coming back to the digestibility reducing capacity of condensed tannins, we know this, but what are we doing about it? It's something that generally gets ignored in most studies of browse quality. But what can we do about it, and what should we be doing about it? Robbins et al. In 1987 and Codron et al. 20 years later made some very pertinent comments, and unfortunately these comments kind of have largely been ignored. They've gone unnoticed. No one's really doing anything about them. But Robbins et al. Was saying that just looking at plant protein as an indicator of plant quality doesn't help, okay? And Codron et al. Were basically saying very bluntly that browse is not more digestible or better quality than grass, which I'm sure, you know, that's the kind of thing that we've been taught, is that browse is better than grass. A few more years later, Wallace et al. Suggested that available nitrogen rather than total nitrogen is important, especially when diets contain tannins. And as far as I know, to the best of my knowledge, there are 3 ways of determining available nitrogen or digestible protein in our browse specimens, our browse samples. But there's a bit of research I feel that needs to be done here to determine which is the best method to use. And there's only 2 papers, recent papers that I'm aware of, where the authors determined digestible protein or available nitrogen, whatever you want to call it. And Wigley et al. Showed very clearly that available nitrogen is way less than total nitrogen in the plant. And interesting to note there is that if you look at the available nitrogen in the top 2 rows of numbers, you'll see there's very little noticeable difference between the acacia species, which we are always told are better quality and more palatable compared to other species. They're all pretty similar, which raises questions. So in conclusion, I'd like to urge people who are either about to start assessing browse quality or are in the process of assessing browse quality to add to the routine analyses that we always do of total nitrogen, ADF, NDF, digestibility, etc., is to include available nitrogen or digestible protein. It's very important to do that in tannin-containing plants. We know this. We should be doing it. Try as far as possible to use species-specific tannin standards. And lastly, and probably the most daunting component to add to these analyses, most daunting to many of us in the room, is to include metabolomic analyses using LC and GC methods or techniques to profile the flavonoids and profile the volatiles. Thank you.
Thank you, Prof.